You know, um, when you picture a tumor, I think it's completely natural to just imagine this growing localized mass of rogue mutated cancer cells.
Right.
Just a clump of bad guys growing out of control.
Right, yeah. That's the classic view.
Exactly. But I want to pose the thought experiment for you today. What if the most dangerous part of a cancer isn't actually the cancer cell itself? What if, the real danger is the innocent, healthy cell surrounding the tumor that the cancer essentially brainwashes to do its dirty work?
It's a terrifying thought, but it completely flips the script on how we view the disease because it forces us to stop looking at a tumor as just a ball of rogue cells and start looking at it as the architect of an entire, rogue ecosystem. It's actively building a neighborhood that protects it.
Yeah, an evil architect. And that brings us to our mission for this deep dive. We are unpacking a truly groundbreaking 2026 paper published in Cell Death and Disease. We're going to explore the hidden battlefield of oral squamous cell carcinoma, or OSCC, to understand exactly how a tumor builds this impenetrable, immune-suppressing fortress. And more importantly, how we might finally be able to breach it.
And just to ground this in a bit of clinical context for OSCC, we know it doesn't just appear overnight, right? It's a stepwise progression.
Right, it takes time.
Exactly. It starts in the normal oral mucosa. Then you often see these precancerous lesions like oral leukoplakia, which, we know that carries up to a 20% risk of turning malignant. And then when it finally crosses that threshold, it becomes invasive carcinoma.
Which is the terrifying part.
Yeah. And the persistent mystery has always been, what exactly happens at that tipping point?
Okay, so let's unpack this, because knowing that a tumor goes from a precancerous warning sign to a full-blown invasion is one thing, but, tracking the actual mechanics of that invasion is a whole different story.
Oh, totally.
To answer that, the researchers behind this paper realized they couldn't just look at traditional bulk sequencing. Taking a blenderized smoothie of tumor tissue and looking at the genetic average just wasn't going to cut it anymore. They needed a high-definition biological map.
Right, because bulk RNA sequencing is great for telling you what ingredients are in the smoothie, but it tells you absolutely nothing about the original structure of the fruit. You lose the context.
So how did they map it?
So they use two incredibly synergistic tools, single-cell RNA sequencing, or cRNA-seq, combined with spatial transcriptomics. With cRNA-seq, you're literally isolating every individual cell and reading its specific genetic barcode.
So you know exactly which genes are turned up or down.
Exactly. You know exactly who is who. But as you were saying, knowing what a cell is doing isn't enough if you don't know where it lives in the neighborhood. The geography matters immensely.
Which is where the spatial part comes in.
Precisely. Spatial transcriptomics acts like a biological GPS. It takes that individual genetic readout and overlays it directly onto the physical architecture of the intact tissue. So now, we don't just know that there's a highly aggressive cell expressing a certain gene. We know exactly where it is sitting relative to blood vessels, immune cells, and healthy tissue.
It's like turning on the lights in a dark room. And using this GPS, the researchers divided the tumor environment into 3 distinct zones. And the way they map this out is just brilliant. Think of it like a medieval siege.
I love this analogy.
Right. So you have the tumor core, which is like the invading king's tent, positioned safely in the back. It's established. It's heavily guarded. Then you have the adjacent stroma, which is the surrounding, relatively peaceful countryside. But the third zone, the invasive front.
That's the bleeding edge.
Yes. That is the actual front line where the battering rams are actively hitting the healthy castle walls.
And what's fascinating here is what they found when they zoomed in on that invasive front. They discovered a very specific subset of cancer cells, a cluster they literally named C4, where a genetic program called partial epithelial to mesenchymal transition, or PEMT, is just highly, highly active.
And PEMT is essentially a plasticity mechanism, right?
Right.
Like these cells are shedding their normal, rigid epithelial characteristics.
Exactly. I mean, normally epithelial cells like to stick tightly together. They form barriers. But these C4 cells are highly plastic. They are down-regulating those adhesion molecules and becoming mobile, flexible, and metabolically prepared to invade deeper into the surrounding healthy stroma.
They're the shock troops.
They are the shock troops, yeah.
So just to pause and recap for you real quick. We have our biological map, and we know the invasive front is swarming with these highly plastic C4 shock troops. But this begs a major question. If you're listening to this and visualizing this battlefield, you might realize something: cancer cells alone can't build a fortress.
No, they really can't.
Right, they don't have the structural tools to lay down dense extracellular matrix. So who are they recruiting at this front line to do the heavy lifting?
And that leads us directly to the discovery of a heavily corrupted local workforce, specifically fibroblasts. Now, as you probably know, fibroblasts are normally the tissue builders and maintenance workers of the body.
Like the construction crew.
Exactly. Their primary responsibility is creating and remodeling the extracellular matrix, the ECM, which is basically the biological scaffolding holding your tissues together.
But at this invasive front, the researchers identified a specific villain, a subtype of fibroblast that just explodes in numbers as the cancer progresses to that highly invasive stage. They called it the COL1A1 positive fibroblast.
And the clinical data attached to this finding is pretty stark. High levels of these specific cells in a patient's tumor correlate with a significantly worse prognosis, like drastically worse.
Wow. And if you want a quick way to anchor this in your mind, just look at the gene name, COL1A1. Think of that rogue fibroblast as a corrupted builder, completely obsessed with pouring massive amounts of collagen to build a literal wall around the tumor.
I like that. COL1A1 pours collagen. It's perfect. And looking at the spatial data, these fibroblasts aren't just randomly distributed. They are physically clustered side by side with those aggressive C4 cancer cells right at the invasive front.
So they're working together?
No, hand in glove. Their job is aggressively organizing this dense extracellular matrix and promoting angiogenesis, building new blood vessels to feed the growing tumor.
But I have to push back here for a second because normal, healthy fibroblasts don't just spontaneously decide to build an enemy fortress, right?
Yeah.
There has to be a mechanism of corruption, a communication channel. What is the hypnotic trigger that forces a healthy structural worker to turn into a rogue COL1A1 builder?
And that is the exact molecular puzzle the researchers set out to solve. And to find that trigger, we have to zoom all the way down to the receptor level and trace this incredible chain reaction. It's essentially a biological Rube Goldberg machine where the cancer cell hijacks perfectly normal cellular machinery to force this transformation.
Okay, I love a good molecular mystery. Let's follow the dominoes. It has to start with a signal from the cancer cell, right?
It does. Those malignant C4 epithelial cells at the invasive front, they secrete a protein called PLAU. That's urokinase type plasminogen activator. So PLAU is the signal propagating through the microenvironment.
Okay, so the cancer is sending out a chemical text message. But the text is completely useless if the fibroblast doesn't have a phone to receive it.
Exactly. And the phone on the fibroblast surface is a receptor called UPAR. But here is the critical twist. UPAR is what we call a GPI-anchored protein. It sits on the cell surface, but it lacks a transmembrane or intracellular domain.
Wait, so it doesn't even reach inside the cell?
Right. It's essentially a broken lock. It can catch the PLAU signal perfectly on the outside, but it has absolutely no physical way to pass the message inside the cell to alter gene expression. It absolutely needs a partner.
Okay. Because without an intracellular tail, you can't initiate a phosphorylation cascade. So what does UPR co-opt to get the message inside?
Well, when PLAU binds to UPR, the conformational change forces UPR to physically recruit and connect with a co-receptor right next to it on the membrane ITGB1 or integrin beta-1.
Integrin, so of course. So ITGB1 is the partner that actually reaches across the membrane into the cytoplasm.
Precisely. This connection activates ITGB1. And once activated, ITGB1 adds a chemical spark. Phosphorylates an enzyme localized just inside the cell membrane called FAK, focal adhesion kinase.
Okay, so now FAK is activated. Does FAK then travel down to the nucleus to change the genes?
No, and this is where the hijacking gets truly devious.
Okay, I'm ready.
FAK stays near the surface, acting as a molecular matchmaker. The activated FAK physically pushes two halves of a completely different receptor system together, TGF-beta receptor 1 and TGF-beta receptor 2.
Wait, really? So PLAU uses an integrin to activate a kinase, just to hijack the TGF-beta pathway. That is an absurdly complex workaround.
It is. It's wild, but it's incredibly effective. Because once FAK forces those TGF-beta receptors to dimerize and bind, it triggers the classic internal domino effect down the SMAD pathway. This cascade ultimately leads to the activation of a transcription factor called CRA3L1, which gets enzymatically cleaved into its active form.
And transcription factors are the keys to the nucleus.
Exactly. That active CRB3L1 travels deep into the nucleus, binds right to the fibroblast's DNA, and forcefully upregulates the COL1A1 gene. The normal fibroblast is officially reprogrammed. The spell is cast.
That sequence is just mind-blowing when you lay it out like that. So, again, to recap for you listening, it's a relay race. Cancer secretes PLAU, which passes the baton to ITGB1, which tags FAK, which forces the TGF-beta door open, sending CRF3L1 into the control room to flip the CO1 equivalent switch.
That's a perfect summary.
But how do we know for sure that this exact sequence is responsible? I mean, biology is messy, right? There are usually redundant pathways.
Which is why the scientific rigor in this paper is just so satisfying. The researchers didn't just map the pathway, they systematically dismantled it in the lab to prove causality. They used highly specific inhibitors to break different links in this chain.
Like breaking the domino line.
Exactly. For example, they used a small molecule inhibitor called VS4718 to block FAK from phosphorylating. They used another drug, PF429242, to block the cleavage of CRF3L1. And in every single isolated instance, when they broke a link in that specific chain, they successfully stopped the fibroblasts from transforming. The hypnosis was completely broken.
That is so elegant. So if we step back and look at the whole picture, the cancer cells have successfully cast this spell, the rogue builders are activated, and they are pouring massive amounts of dense collagen at the invasive front. But if you're listening to this and thinking critically about the immune system, you might be asking a big question right now.
Why does a physical wall even matter?
Right. We have cytotoxic T lymphocytes, CD8 positive T cells, that are specifically designed to infiltrate tissue and kill cancer. Why don't they just squeeze through the matrix and break the siege?
And if we connect this to the broader concept of tumor immunology, this brings us to the most insidious finding in the paper. It's not just a physical barricade. It's what the researchers call a dual immunosuppressive niche. Let's look at what happens once that extracellular matrix is heavily remodeled by these rogue COL11A1 positive fibroblasts.
Okay, lay it on me.
So, yes, the matrix becomes incredibly dense and physically alters the microenvironment, making it mechanically difficult for killer T cells to penetrate. But the wall is also chemically toxic to the immune system.
Chemically toxic? In what way?
The dense ECM, combined with specific cytokine signals secreted by those corrupted fibroblasts, actively acts on healthy immune cells that approach the border. When healthy CD4 positive helper T cells arrive at the scene, the microenvironment literally forces them to differentiate into regulatory T cells, or TREGs.
Oh, wow. Which is a brilliant and terrible irony. Because in a normal immune response, TREGs are the body's peacekeepers, right? We need them to enforce immune tolerance, to calm the system down after an infection is cleared so you don't develop autoimmune disease.
Exactly. You need TREGs so your body doesn't attack itself. But here, the tumor exploits that exact tolerance mechanism. It essentially recruits the body's own peacekeepers and uses them as bouncers. These induced TREGs actively suppress local immune responses, shutting down the dendritic cells and effectively paralyzing and locking out the cancer-killing cytotoxic T lymphocytes.
So, to summarize this dual barrier for you, the tumor uses the body's own structural workers, the fibroblasts, to build a dense physical wall, and then uses those workers to recruit the body's own immune peacekeepers to chemically guard the wall.
That is the dual barrier in a nutshell, a physical fortress of collagen guarded by a chemical moat of immunosuppressive TREGs. They've weaponized our own biology against us.
Which leaves us with a rather massive pragmatic question. If the tumor has built this perfect, multifaceted fortress out of our own corrupted cells, how on earth are oncologists supposed to breach it? Because traditional drugs don't seem like they'd even scratch the surface here.
This is where the paper pivots from being a terrifying biological mapping exercise into offering a very real, tangible ray of hope. Because by mapping that massive Rube Goldberg machine, they found an Achilles heel.
Okay, where do we strike? Because if I'm looking at the pathway, my first instinct is to go after the source. Do we try to block the cancer cells from secreting that initial PLAU signal?
It's a completely logical thought, but practically it's incredibly difficult. Remember that dense collagen matrix? It makes it exceptionally hard to get neutralizing antibodies or small molecules deep enough into the tumor core in high enough concentrations to target the PLAU secretion directly.
The wall blocks the medicine?
Right. And furthermore, cancer cells are notoriously genetically unstable, right? If you target them directly, they often mutate and find a workaround. But remember the relay race. The fibroblasts are genetically stable. We can target the receptor on the healthy turned rogue cells. We can target the broken lock's partner, ITGB1.
Wait, let me push back on that for a second. Integrins, specifically integrin beta-1, are ubiquitous. I mean, they are everywhere in the body, facilitating normal cell adhesion and extracellular matrix communication. If we just systemically carpet bomb a patient with ITGB1 inhibitors, aren't we risking massive off-target toxicity? Won't we destroy healthy tissue remodeling?
That is the exact right question to ask, and honestly, it's a major hurdle in integrin-targeted therapies. However, in their preclinical mouse models, the researchers didn't just blindly administer a systemic integrin blocker. They designed a localized rescue experiment. They utilized a highly specific anti-ITGB1 monoclonal antibody treatment combined with standard immunotherapy, specifically an anti-PD-1 drug, which acts to take the brakes off the immune system.
Okay, so it's a combination approach, breaking the wall and simultaneously arming the troops. What were the results?
The results were a breakthrough. By blocking ITGB1 at the tumor site, they intercepted the PLAU signal. The downstream cascade stopped. As a result, the population of COL11A1 positive fibroblasts plummeted.
Yeah, because the hypnosis was broken.
Exactly. Without the rogue builders maintaining it, the physical collagen wall began to degrade. Without the chemical signals from the fibroblasts, the immunosuppressive tregs dwindled. And suddenly, that anti-PD-1 immunotherapy, which previously couldn't penetrate the fortress, was able to rush in with cytotoxic T cells and dramatically shrink the tumors in the mice.
That is incredible. Just completely dismantling the ecosystem. But here's where this paper gets really interesting for human patients who are walking into clinics right now. The researchers realized that these TOL1A1 positive fibroblasts and the ITGB1 receptor pathway aren't just potential future drug targets, right?
Yeah.
They are incredibly accurate biomarkers available today.
Right. They expanded their view way beyond just oral squamous cell carcinoma. They mined massive clinical databases across multiple different types of solid tumors. They looked at patient data for triple-negative breast cancer, colorectal cancer, and non-small cell lung cancer.
And they found a universal truth across all of these different tissues.
They did.
Patients who naturally had low levels of these rogue COL other MA1 fibroblasts in their tumor microenvironment responded beautifully to standard immunotherapy. Their immune systems were able to do the work. But patients whose biopsies showed high levels of these specific fibroblasts, they overwhelmingly did not respond. The fortress was simply too strong.
And this completely shifts how we might approach treatment pathways in the immediate future. I mean, if a patient is diagnosed with a solid tumor, before we put them through grueling cycles of expensive and exhausting immunotherapy, we could simply stain a biopsy for COL1A1. By mapping the neighborhood, doctors can predict with high accuracy whether standard immunotherapy will actually work.
And if the patient have high COL1A1 levels, the doctor knows they need to bring in a combination therapy.
Yeah.
Perhaps something targeting the stroma or the integrin pathways like we talked about, to break the walls down first,
Right.
rather than wasting precious time on a drug that will just bounce off the fortress.
Exactly. It raises a fascinating philosophical shift in oncology. We spent the last several decades trying to develop drugs that act like highly targeted snipers, obsessively trying to kill the mutated cancer cells directly. But this paper proves that a solid tumor isn't just a solitary rogue entity. It absolutely requires a hypnotized, actively collaborating microenvironment to survive.
Which means the future of oncology might not just be about finding better ways to kill cancer cells. It might be equally focused on learning how to deprogram the healthy cells. If you break the hypnosis and remove the support system, the fortress crumbles from the inside out, exposing the cancer to the immune system we already have.
It's a powerful reminder that sometimes the most effective way to win a siege isn't to repeatedly bash your head against the reinforced castle wall. It's to figure out who is supplying the mortar and convince them to stop building.
I love that. So next time you hear about a massive cancer breakthrough in the news or read an article about a new miracle drug, I want you to ask yourself a question based on what we explored today. Are they attacking the castle directly, or have they finally figured out how to fire the rogue construction workers? Think about it. Thanks for joining us on this deep dive. Keep asking those hard questions, keep exploring the hidden mechanisms around you, and we'll catch you on the next one.